Morning students.
Morning, sir.
Today our pathway is about TCA cycle. The name itself indicates that TCA means tricarboxylic acid cycle. Here tricarboxylic acid means it contains the compound which is entering into the TCA cycle contains 3 carboxylic acids. For example, citrate is the compound which is entering into the TCA cycle, which contains 3 carboxylic acid. That's why we are calling it as a tricarboxylic acid. It is also be called as Krebs cycle. Why means it is given by the scientist known as Hans Krebs. That's why we are calling it as a Krebs cycle. And it is also be named as citric acid cycle. Why means the first intermediate entering into this pathway is a citrate. That's why we are calling it as a citric acid cycle. And this TCA cycle is a pathway which is the second part of glucose oxidation. Why I am telling is, in the first part of the oxidation of glucose, glucose is converted into the pyruvate. In the second part, the pyruvate entering into the TCA cycle. Here the remaining metabolites are oxidized. That's why we are calling it as the second part of the TCA cycle. So this is the pathway which is occurs in mitochondria. This is a pathway occurs in mitochondria, especially in the mitochondrial matrix. Especially in this is the mitochondria. Here it is the matrix. In mitochondrial matrix only TCA cycle is occurring. Why means all the enzymes of TCA cycle are present in mitochondrial matrix only. That's why TCA cycle is occur in the mitochondrial matrix. And there is another reason why it is occurs in the mitochondria means the reducing equivalents like NADH plus H plus and FADH2 produced are entering into the electron transport chain which is present on the inner side of the inner mitochondrial membrane. They can easily enter from matrix to the electron transport chain. That is the reason why the TCA cycle is occurring in the mitochondrial matrix. Now we will enter into the pathway that is the end product of glycolysis, which is a pyruvate, which is the connecting link between the glycolysis and TCA. Okay. In the pyruvate, which is formed in the, for example, it is a cell. This is the cytosol. This is the cytosol. Glycolysis occurs here. That is, glucose is converted into the pyruvate. And this pyruvate which is formed in the cytosol will enters into the mitochondria present in the cytosol. Here where TCA cycle will be occur. After entering the pyruvate into the mitochondria, the reaction, the first of all happening is pyruvate converted into the acetyl CoA. Pyruvate converted into the acetyl CoA. In the presence of enzyme pyruvate dehydrogenase. As you know that dehydrogenase means it is removal of hydrogens. That is, 2 hydrogens are taken from the pyruvate by the NAD to converted into the NADH plus H plus. In the presence of CoA enzyme to form acetyl CoA. Being pyruvate is a 3-carbon compound, but acetyl CoA is a 2-carbon compound. Here, one CO2 is released. One CO2 is released. That's why 3-carbon is converted into the 2-carbon acetyl CoA. And this is reaction is the connecting link between the glycolysis and TCA. This TCA cycle is mostly occurred in aerobic organisms only. Why means anaerobic organisms doesn't contain, that is especially bacteria doesn't contain cell organs like mitochondria and other things. And this pathway occurs only in the presence of oxygen only. For example, if there is a cell, glucose converts to pyruvate. If the oxygen presence is there, then only pyruvate will enter into the mitochondria for the TCA cycle. If there is no oxygen presence is there, pyruvate will never enter into the TCA cycle for entering into the citric acid cycle. For that reason, that is pyruvate will enter into the TCA cycle, which is a connecting link between this complete oxidation of glucose. After connecting link between the pyruvate to acetyl CoA, the acetyl CoA, which is a 2-carbon compound, which will combine with the first intermediate, that is oxaloacetate. This oxaloacetate is a 4-carbon compound. Oxaloacetate is a 4-carbon compound and acetyl CoA is a 2-carbon compound. 4 carbon plus 2 carbon form 6-carbon compound citrate. That's why we are telling it as a citric acid cycle, which is the first intermediate in the TCA cycle, and it is the containing the 3-carboxylic acids. That's why we are calling as a tricarboxylic acid cycle. Okay. The citrate so formed in the presence of enzyme aconitase to form cis aconitate. Here a removal of water molecule will happen, and this cis aconitase is an intermediate between the citrate and isocitrate. This is very fraction of second step. And this cis aconitate in the presence of same enzyme, aconitase, here addition of water molecule will happen to form isocitrate. And this isocitrate, it is also the 6-carbon compound, and isocitrate also be the 6-carbon compound. Isocitrate in the presence of isocitrate dehydrogenase. As already I told that when the dehydrogenase will come, there is a removal of hydrogens. So NAD, isocitrate, NAD takes 2 hydrogens from isocitrate to form NADH plus H plus to form oxalosuccinate. Here again it is a fraction of seconds intermediate. So isocitrate dehydrogenase is the same enzyme will convert oxaloacetate to alpha ketoglutarate. Here oxalosuccinate is also be the 6-carbon compound, but alpha ketoglutarate is 5-carbon compound. So as already told that it should be there is a decrease of carbon means compulsory there is a removal of CO2. So that's why 6-carbon compound is converted into the 5-carbon compound alpha ketoglutarate. Alpha ketoglutarate in the presence of enzyme alpha ketoglutarate dehydrogenase, where again we have indicated the dehydrogenase, where again NAD takes 2 hydrogens from alpha ketoglutarate and form to NADH plus H plus. Okay. And here again there is a removal of CO2. Alpha ketoglutarate is 5 carbon compound. Removal of CO2 means it is converted into the 4 carbon compound. That is, it is converted into 4 carbon compound succinyl CoA. Addition of CoA into the reaction. That's why we are forming the succinyl CoA. And this succinyl CoA in the presence of enzyme succinate thiokinase, as already indicated that when there is a kinase reaction will happen, compulsory there is an ATP or GTP or any involvement will happen. That's why here again GDP converts to GTP. Okay, succinate CoA. One inorganic phosphate will enter into the reaction. GDP takes place this inorganic phosphate to form the GTP, and COSH that is added here in succinyl CoA is removed in this reaction to form succinate. Okay, and this succinate in presence of succinate dehydrogenase, again dehydrogenase reaction is taken place. That's why it takes 2 hydrogens from succinate to form FADH2 to form fumarate. Here succinate is 4 carbon, succinyl CoA is 4 carbon, fumarate also be the 4 carbon compound. Fumarate later removal of water molecule to form the L-malate. It is very less time reaction, that's why the same reaction is, that is fumarase. The enzyme here is fumarase, which will convert the fumarate to malate. And malate in presence of malate dehydrogenase, again dehydrogenase reaction, NAD takes 2 hydrogens from malate to form NADH plus H plus to form oxalosuccinate. That is the intermediate which we have started in initial will regenerated, and this oxaloacetate will take another acetyl CoA which is coming from the glycolysis, which will combine with them to form citrate, and the cycle will continuous like this. That's why we are calling it as a TCA cycle. Okay, and this TCA cycle is meant for the production of ATP, meant for the production of ATP generation. Why means glycolysis, the production of ATP through glycolysis is only 8 ATP, which is in very less amount, but those cells contains the, that is higher organisms, they are all eukaryotic organisms having the cell organelles present in the cytosol. This pathway will enter into the TCA cycle and will form more ATP. How this more ATP will form, we will see. There are 2 types of phosphorylations, as you all know that 2 types of phosphorylation. One is oxidative phosphorylation, another one substrate level phosphorylation. Oxidative phosphorylation means the intermediate, that is NADH, NAD converts to NADH plus H plus, NAD converts to NADH plus H plus, FAD converts to FADH2 plus S, and this NAD converts to NADHS plus. All these enter into the where? ETC. Where? ETC, electron transport chain. All these formed hydrogens, NADHs, enter into the electron transport chain for production of ATP, for the production of ATP. As we know that one NADH plus H plus enter into electron transport chain gives how many ATP? 3 ATP. And one FADH2 enter into the electron transport chain gives how many ATP? 2 ATP. Okay. In this way, calculate how many ATP are formed here. How many NADH are there? In this pathway, how many NADH are there? See clearly. One is here, one is here, one is here. So, how many ATP will you get? 3 into 3 equal to 9 ATP. And FADH2 is 1 FADH2. That's not 1 into 3, 2 ATP. So total 11 ATP. But in TCA cycle we are getting 12 ATP. That is, there is one substrate level phosphorylation here. GDP converts to GTP. GDP converts to GTP. So one substrate level phosphorylation means one ATP is added to form total 12 ATP. 11 plus 1, 12 ATP is formed. This is for one pyruvate. This is for one pyruvate convert to N-acetyl-CoA which is enter into the TCA cycle to form 12 ATP. How many pyruvates are we are having? From glycolysis to pyruvate, how many pyruvates are formed? Glucose is 6 carbon, pyruvate is 3 carbon. How many pyruvates we are forming? 2 pyruvates. For one pyruvate which enters into the TCA cycle as acetyl-CoA and form this reaction to form 12 ATP. We are having 2 pyruvate molecules, that is 2 into 12. That means 24 ATP are formed. How many ATP? 24 ATP are formed in the TCA cycle reaction. And in glycolysis, how many ATP we will get? 8 ATP. 12 plus 8, sorry, 24 plus 8, 32. But in glycolysis, we are getting 38 ATP. Where the other ATP are coming? Where are the other 6 ATP? That is from connecting link. Here is pyruvate dehydrogenase. We have calculated this one. We have calculated the glycolysis one. But we have not calculated this NADH, which is a connecting link. This is also formed. NAD converts to NADH plus H. One pyruvate, one acetyl-CoA gives one NADH. Two pyruvate gives two acetyl-CoA means two NADHs plus. That is, we are having 2 into 3 entering into ETC to form 6 ATP. Now 32 plus 6. How many? 38 ATP which are formed from glucose to TCA cycle. That is complete oxidation of glucose. Complete oxidation means glucose is converted into the CO2 plus H2O. This is called as complete oxidation of glucose. Only oxidation of glucose means glucose is converted into the pyruvate. Complete oxidation means this pyruvate also enters into the TCA cycle to form CO2 plus H2O. That is, we have started with the 6 carbon compound citrate and finally we are getting the oxaloacetate which is a 4 carbon. Where the other 2 carbons? Which was released in the form of CO2. One CO2 released here, one CO2 released here. That's why we are telling it as it's a complete oxidation gives as CO2 plus H2O. 2 CO2 are released. And this total pathway is happening due to the main reason for the production of ATP. Total production of 38 ATP. And TCA cycle is also having more importance, like it is the most important cycle for formation of large number of intermediates. That means, and that phenomenon is called as anapleursis. Anapleursis. Anapleursis means filling up of intermediates of TCA cycle. Filling up of intermediates of TCA cycle are called as anapleuritic reactions. Means, why we have to fill up the intermediates of TCA cycle means, TCA cycle contains large number of intermediates which are gone to other reactions when the cycle is happening, but the pathway should not be disturbed. For that, there are some reactions known as anapleuritic reactions which will happen and fill these intermediates. For example, a reaction for formation of glutamate amino acid, alpha ketoglutarate is required. It is gone through the reaction. But what happened, the TCA cycle pathway which has stopped in the pathway. For that, anapleursis, what will it do? It fill up the alpha glutarate from other reaction to form the alpha glutarate. Then cycle again will continue. Those reactions are called as anapleuritic reactions. Okay. And this pathway having two phases, like it is a amphibolic pathway. What pathway? Amphibolic pathway. Why it is amphibolic pathway means, in this TCA cycle we are having two types of reactions. Anabolic reaction, that is forming up of compounds are synthesized, and catabolic reactions, compounds get breakdown. Okay, anabolic reactions are there, catabolic reactions are there. That's why we are calling it as a amphibolic. The only pathway, major pathway amphibolic in nature is TCA cycle.